Hello, my name is Peró and I come from Sant Pau Hospital in Barcelona. In the first place, I would like to thank the scientific committee for selecting our work. We don't have any conflict of interest in relation to this presentation. We present our experimental study in which we analyze the biological properties of a human cellular dermal matrix implanted in a rabbit vaginal and abdominal model. Mesh reinforced repair is currently still a standard procedure in pelvic reconstructive surgery. Prosthetic materials are usually synthetic and unabsorbable. As synthetic mesh materials are associated with the risk of severe complications, there is a growing interest in finding better materials that help improve outcomes. Our research group studied the properties of a biological graft material, a human cellular dermal matrix, HADM, to be used in reconstructive pelvic surgery by testing it in an animal model. HADM is an extracellular matrix developed by Tissue Banc, in which the starting material is human cadaveric skin that undergoes the cellularization process, leaving the extracellular matrix intact. 20 New Zealand rabbits were randomized in two groups. 10 received ADM grafts, while the remaining 10 were implanted with polypropylene mesh. Each rabbit had 4 segments implanted, 2 into the abdomen and 2 into the vagina, in the submucosal layer. The graft segments were removed 180 days later. Rabbits were monitored until euthanasia. After sacrifice, grafts were explanted to perform different analyses: microscopic findings, biomechanical properties, immunohistochemical and histological properties. Here we can see some pictures about the surgical procedure. Here we can see the abdominal approach, and here we can see that the vagina and the vaginal entrance, and that's the anus. And here we can see some pictures about the implantation. That one graft in the anterior wall of the vagina, and here it's the second graft, the second segment implanted in the posterior wall of the vagina. And here we can check the final result in the implantation surgery in abdominal approach. Rabbits were successfully burned until euthanasia. One control group subject died due to causes other than surgery or implant placement complications. Greater abdominal findings like extrusion, mesh, or wound infection were seen during the follow-up in the control group against the experimental group. The macroscopic study of explants showed a significant increase of vaginal mesh extrusion in the mesh group. However, macroscopic degradation of the vaginal graft was more common in the experimental group, whereas in the control group, the vaginal mesh was identified in 100% of the individuals. Regarding the biomechanical study of explants, we regret to explain that stiffness and elasticity were worse in the experimental group, especially in vaginal location. Both polypropylene and HADM grafts showed cellular infiltrates corresponding to an inflammatory foreign body response. HADM integration was complete, especially in the vaginal location. This project could provide potential therapeutic, scientific, and technical benefits that could have relevance and applicability in the field of regenerative medicine, and specifically in the field of urogynecology. These results offer effective therapeutic options based on the use of human tissues in clinical practice to improve current therapies which are suboptimal. We demonstrate a need for optimization of explant collection and processing protocols and improved knowledge of their characteristics, behavior, and properties to be applied to reconstructive and therapeutic purposes. Finally, our results can be shared with the field of urogynecology. So, we can conclude that New Zealand white rabbit is a good model for assessing materials to be used as graft for pelvic reconstructive surgery and vaginal surgery. Animals are easily managed during the procedures, including surgical intervention and vaginal mucosa probe. Finally, HADM implantation is associated with fewer clinical complications, as well as better macroscopic tissue integration, but with poorer biomechanical properties 6 months after implantation in the vagina compared to polypropylene mesh. Thanks for your time. I am at your disposal for anything you need.